the foods. Okay, so this is milkfish, mango, sliced beef. Yeah, very nice. Wonderful. And I think you all know this. Yeah, yeah, this is the great invention of Taiwan. All right, so uh, I really feel quite familiar with you because in our university and even in our college, we have lots of Indonesian students. Yeah, example, among our 194 undergrad students, 36% of them are international students. Oh. And most are from Malaysia, but we do have lots of students from Indonesia. Right? We have 26 Indonesian students in our college, and you can see they're having fun here. They went to hot pot, okay, and they have a very nice tradition, which is the second year Indonesian student will treat the first year Indonesian student. Okay, so if you ever go to Taiwan, especially National Capital University, you'll be treated by your seniors. Okay, and these are some of our graduate students. We have 6 Indonesian students studying their master and PhD. Yeah, and actually, this is one of my college students. He's now doing the spatial topics in my lab. He's quite talented. Yeah, we have some internship program. So he did his internship in Academia Sinica, which is the highest research institute in Taiwan, and he got a second place. Yeah, it's very talented student. So I have very good impression of you. Yeah. And he now got the exchange student opportunity. He's going to I think Europe for a semester. Yeah, one is so supported by our college. Okay. Yeah, so we do have some exchange student opportunities between NCKU and many other universities all over the world. Okay. So if you ever visited or NCKU or study in NCKUs, you'll have chance, yeah, to move on to other countries. So, all right. So, yeah, you're welcome to Taiwan, but you don't come to Taiwan just for the food, right? You want to study, you want to gain some skills. And so this picture shows, summarize what we are doing in our college. So you can see that we have professors studying tomato. Yeah, she's studying the sugar transport mechanism in tomato. Okay. And can you guess what this is? Algae. Okay. Yeah. So we also study microalgae. Yeah. And one of the applications were quite interesting. Have you ever heard of DHA? DHA, that kind of fatty acids. DHA, very good supplements. Yeah. Very good for your brain. That's why you don't know. Yeah, so this particular microalgae is very amazing. It can store up to 69% of its body weight of DHA and fatty acids. Okay, so we are trying to coding the genome and trying to see what's going on, how it is able to tolerate such a high concentration of DHA, yeah, in its body. All right, so we have some other research topics, for example, the shrimp. Okay, so one of our professors is studying shrimp. She is studying the white spot virus in the shrimp. Okay, so she's studying the mechanism of this white spot virus infection. Okay, so after lots of work to figure out the fundamental mechanism, this bacteria, this virus infection. After that, figure out a way to prevent the infection. Okay. And so she's able to raise the shrimp to a very large size. Okay. This big, okay, this big. And if you go to NCKU, you can buy the shrimp from our souvenir shop. Yeah. Very tasty. Just put it in the oven. Yeah, just steam it a little bit. Yeah, and then you can show. Yeah, the research findings. Okay. So I'm trying to translate fundamental research to industry. Example, our thing is working on information study. So we found a compound that can reduce the information. So he just thought that, oh, perhaps I can mix the ingredients to the cosmetic products. Okay, so for example, if you are young, I think most of you are young, maybe you have pimples on your face. It's kind of information condition, right? So if you can try the cosmetics, lotions with anti-inflammation function, that would be nice, great, right? So again, you can, if you come to NCKU, you can find that in our souvenir store. Okay? You can buy the cosmetics. All right, so we have many different research topics. Actually, most of the topics are also studied by your faculty members. Yeah, here. So, you are in a very good environment. So. And a little bit self-introduction. I actually never studied biology. Yeah, in my high school. Okay, because at that time we are allowed to skip biology if you're not going to medical schools. Okay, if you want to go to engineering schools like physics, yeah, like chemical engineering, some electronic engineering, then you don't need biology. So at that time I was lazy. Yeah. So one subject less. Okay, it's easier. So I didn't study biology. And I did my bachelor degree in physics and also my PhD in physics. Okay, so this is my thesis topic of my PhD is statistical physics of RNA secondary structure. Okay. And why I picked this is because when I went to Ohio State University, where I did my PhD, this is my advisor. Yeah, he came and gave a lecture about this RNA secondary structure. Okay. And showed me that this RNA secondary structure, if you want to describe the properties, this structure, you can use a formula, yeah, to describe it. Yeah. And I was like, oh, I used to be a physics student. I love formula. And it was surprising that I can use a formula to describe a biological molecule. Wow, it was like fantastic to me. So I ended up studying in his lab, and I love this formula a lot. Yeah, and after I graduated, I was looking for a postdoc. Okay, so because I was studying this RNA secondary structure, I thought, oh, let's add it to biophysics. I still want to find a job in physics, field of physics, but related to biology. So this Professor Boris Streiman is a physicist in UC Santa Barbara. He's doing biophysics. And I thought that, oh, it's great. Yeah, this is my target. I try to call him. And then, so we chat a little bit in the forum, blah, blah, blah. And then he said, okay, you can come. Okay. So I went to UC Santa Barbara. I imagined myself to stay in the office with blackboards and people writing chalks. Yeah, blackboards. Okay. I just learned that you are still writing in chalk. Yeah, I love this kind of stuff a lot. That was in 360 pound. Yeah, but now I do this only. I use only PowerPoints like nowadays. Okay. But it turned out that when I went to UC Santa Barbara, yeah, Boris Streiman was actually looking for a postdoc for his friends. Yeah, Ken Coase, who is a biologist, Neuroscience Institute. And so I ended up staying in Ken Coase's lab, okay, and doing bioinformatics. Okay, yeah, that's how I started my bioinformatics career. This is what my lab, how my labs look like. Just sharing with you like your career path is usually full of surprise. You think that you are going to be a biologist, maybe like 10 years later you are AI engineers. Okay. So try to stay open-minded, try to learn something. Yeah. Uh, it's can be beyond your okay. And then when I came back to Taiwan, I went to Academia Seneca, which is the highest research institute. And my advisor told me, hey, Tsunglin, you should start working on this so-called next generations. Okay. And since then, I was doing data analysis on next generation sequencing data till now. Okay. Yeah. So this is my sir. Yeah. He guided me. Yeah. Through my life. Yeah. So, and today I'm going to tell you some story about this machine. Okay. This is the so-called next generation sequencer. And before that, I want you to guess how much was this sequencer when around 2010. This is the first version of the Illumina sequencing machine, genome analyzer. So how much do you think it is at that time? Yeah. Yes. Maybe a billion. Dollar. What kind of dollar? Is it like US dollar or US dollar? How much? How great is he? Yeah, I prepared some gifts. Yeah, it's almost $1 million. You're right. It's like $0.8 million. Okay. Yeah. So this is my machine. $0.8 million around 2010. Okay, around 15 years ago. And I want to say that when I went to NCKU, the professor next to my door, yeah, he bought this one. He was very rich. Yeah. So at that time, I was teaching bioinformatics class, and then I show my students, let's go to the professor's lab. Let's check with this sequencer. Yeah, he enjoys life better than me. He just stays in the air conditioner room with 24 degrees Celsius all year round. Okay, sitting at a table like in shaking thing. So yeah, he really enjoyed his life. And but like uh 5 years later, yeah, professor next to my door shut down this machine. Okay? Shut down this machine. And imagine that you spent like nearly $1 million after 5 years, shut it down. Just like throwing this money, yeah, away. Yeah, garbage. Okay. Why is that? Why do you think he shut down the sequencer? Yes.
Because it ran out of breath, like apa namanya? Because the machine is already apa namanya? Enggak lah, apa namanya? It's already apa namanya?
Don't worry, I still have a candy for you.
Wait. The technology in the machine has is outdated compared to other machine that has been developed at the time.
I have to give you two. Very good, very good. So so it was outdated. Yeah. And do you know what outdated implies? Why when something is outdated, like my iPhone is still iPhone X. I'm still using it. Okay. But why an outdated machine? Yeah. Yeah. You don't want to use it. Why is an outdated machine? If a machine is outdated, you don't want to use it anymore. And that's because when new machines comes out, the cost of sequencing decrease half. Okay, so suppose you need to spend $100 using the original machine, and using the next generation, the next version is 50, the next one's 25, next one's like 20, 15. Yeah, $15. So that's why that professor shut down the machine and he outsourced the sample. Okay, so this one is actually called, I prefer to call it now generation sequencer. Okay, and it still can be quite expensive. Not that expensive as it just came out. Yeah, still expensive. Okay, so this is the number of sequences in 2015. Okay, this is a world map of NGS. People call it next generation sequencing, abbreviated as NGS. Okay, and you can see that there are lots of, like, these are like 3-digit numbers. And so, for example, can you figure out where it is here? Yeah, the test of your geography knowledge. Yeah. Canada? Not really.
New England?
New England? No.
New England, like near Maine.
Huh?
It's like near Maine.
Near Man?
Northeast. Yes.
I'm not sure about this. How about this? Yeah. One of these two. Yeah. Where do you think it is?
York.
New York. Who said New York? Yes.
Yeah.
It's right. Okay. New York. It's somewhere near Harvard and MIT. Okay.
Oh.
So I bought like hundreds of such sequences. Yeah, in their university. Okay. And it's like two big hubs of NGS sequencer in China. Okay. And this one, yeah. What about this? Yes, everybody knows. Yeah, good job. Okay, it's good. Okay. Yeah, so imagine that one single machine costs nearly $1 million. How much does the company earn? Yeah. So if you somehow lucky enough to buy the share, that share of the company, you're a rich guy right now. And I wasn't so lucky. Okay. Yeah, I should buy the stock. Okay. All right. So there is a reason why these NGS sequences are so popular, because they generate sequences like this. Okay. So you can see that there's lots of sequence, CCTAC. And it's pretty long, like it's 300. And the amazing thing is that it can generate like millions of such millions. Before NGS, do you know how many sequences? Before NGS, the gold standard of sequencing is called Sanger sequencing. Okay, and guess how many sequences you can get? Yeah, by doing Sanger sequencing. How many? Thousands. Fewer. Fewer. 50. Fewer. 30. Fewer. 2. Fewer. Just one. If you do single sequence, you get only one sequence at a time. Okay, just one sequence. And this NGS, it generates million sequences, yeah, at a time. Okay, so now you can start to appreciate, yeah, its value, right? It's like a million times faster compared to the old standard single sequencing. Okay, and so imagine that you have such kind of data, yeah, NGS sequences, like millions of them, And so what's next? Once you have this data ahead, what's next? Next thing to do. Okay. So the next thing to do is to extract meaning from these sequences. Okay. That's what we say to decode the genome. Code the genome. Right. So let me give you some examples of what I mean by extracting meaning from the sequences. All right, so imagine that you have such a machine and you are able to do sequencing of the genome of many different species. Okay, so the big question will be, what can we learn from sequencing one of the organisms or many organisms in different environments? Okay. What can we learn from them? So let's make it concrete and try to focus on as human. Okay. Imagine that you are sequencing the human genome. What can you learn from sequencing the human genome? Okay. So now discuss with your neighbors. Come up with a potential application that you can learn. Yeah, from sequencing human genome. Okay. Yes.
You come up with a spell for cancer. I don't know.
Yeah. About cancer. Okay. So what about cancer? Yeah. If you sequencing, if you sequence your genome, what about cancer? Yeah. Actually, this is the right answer. You know, one of the right application. If you want to treat cancer, one way is through the so-called sequence, is to do sequence. Let me jump to the application. Okay, so this is the a field of application called precision medicine. Okay, precision medicine. And now I want you to read the figure and then tell me what it means. Okay, yeah, biology students should be good at illustrating figures. Okay, yeah, yes. You okay. You haven't got the candy yet.
To illustrate the picture, right?
Yeah, that's right. How do you explain this picture? What's the idea of precision medicine?
So what I can,
How about you use the mic?
oh yeah, from what I can get from precision medicine, if we can learn individual genomes, so we can give them medicine that can more effectively treat their conditions and health problems. Whereas with traditional medicine, it's a one size fits all. Some medicines might work less for people, some medicines might work more, but with personalized medicine, you can get something that's tailored for your needs, thus completing your issue much faster.
Wow.
How do you learn it? Where do you learn that from? Or you just know it? Yeah, from the blue. Out of the blue.
It's not.
Both.
Usually I watch YouTube a lot.
You watch what? YouTube. Oh, okay. Watch YouTube more. Really?
I also watch a lot of scientific content, biological content, especially in biological field of more towards health and medicine. I do have quite the interest in that.
Oh, what kind of contents? Like different biological material.
Like for example, the progress of like medicines and research upon infectious diseases, like ways of treating tuberculosis and all
Okay.
that.
Yeah, so you read some news,
On progress of science, but
Progress of Science, there's a magazine, or
No.
website, or
more
channel,
of YouTube channels.
YouTube channel.
Yes, that's
YouTube.
specialized.
Okay, YouTube are your best friend. Yeah, watch YouTube more. Okay, all right, so that's one very important application. So now in the clinics, the most applicable scenario is cancer. Okay, and because have you ever heard of targeted therapy? Yeah, targeted therapy is pretty expensive. Yeah, I know the price. Yeah, in terms of New Taiwan dollar, converted. Okay, so how much do you think if you want to take the targeted therapy for treating your cancer? how much you should pay a month.
$500,000?
No, no, a month.
No, 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 no.
Yeah. I'm rather die. $11 million. Okay.
On what country?
Huh?
It really depends on where you live.
Yes.
Some country charge you more for the same.
For example, America, like they can utilize their health system.
Yeah.
In Taiwan, you pay around 3,000 US dollars. 3,000. But that's already a lot.
Yeah.
Yeah, because most Taiwanese they make like only 50, like 1.5 thousand month, because it's like twice their monthly salary. Okay, so targeted therapy is pretty expensive. So before you take the treatment, you need to make sure that it work. Right? So how do people make sure that targeted therapy will work? How? Sequencing. Okay, sequencing. Right? So you sequence your genome and 10 lots of this, and then we try to convert it to something. Yeah, like that. Okay. So you'll find that within your genome, or actually within the genome of your cancer cells, it's not your normal cells, it's your cancer cells. Find that at some position of the gene, there's a picture. Okay? And these determines which kind of targeted therapy you need to treat your cancer. Okay? So sequencing is very important, right? And also, you mentioned that you 10 NGS sequences. All right. So usually, what's the first thing that you do? Okay. So the first thing that you do is that yeah, you want to figure out our human genome. Okay. So our human genome, it's good to always know some numbers. You know the size of our, how many bases are there in our human genome. Huh? 3 billion. Okay, so it's a very huge, and along our human genome, not all the genomic segments are within a gene. Okay, you counted the segments of protein coding genes. What do you think? What's the percentage of that protein coding genes?
15%.
50? Fewer. 2%. It's a little bit more. Yeah. 5%. It's like between 2 to 5%. Okay. Yeah. So between 2 to 5%. So you can see that the gray area here, they're not genes. They're not protein coding genes. Okay. So if you ever learn this fact, yeah, do you feel anything? Do you have any feeling for this fact?
Much smaller than expected.
Huh?
Most smaller I didn't expect it to be that small.
Huh?
The coding region is very smaller than expected.
Yeah, it's very small, right? So that implies that 95% of our genome is mapped. They are not protein coding. They don't code for proteins. Okay, so what do? 5% of our genome, what do they do? Are they junk? They are junk. Why do we carry 95% of junk in our genome? This is very interesting. Very interesting. It turned out that there are some so-called non-protein coding genes within the genome. Okay. So, for example, small RNA, long non-coding RNA, many different kinds of non-protein coding genes exist in the genome. Okay? The people used to think that our genome is full of junk, but now they started to learn that there are some people call them dark matters, but people still to realize the value, the meaning of this. So this is something that we can learn, yeah, by sequencing the human genome. Okay. And in order to do that, you need to first know the region of protein coding gene, right? You want to know the function of protein. And you know what? Yeah, you can do that by writing computer codes. You can write a program to predict where in the genome is a gene for coding protein. Isn't it amazing? And the program is able to tell, yeah, whether this part for protein or not, whether it's a protein coding gene. Okay, so that's one important topic, bioinformatics. Yeah, and so amazing, yeah, amazing to me. Yeah, where the program and this program is able to decode the meaning of gene. Okay. And then just like I said, you can figure out whether your genome carry a mutation at a certain position and whether the mutations will affect your phenotypes. Right? So when I say phenotypes, this is one of the example. Yeah. Have you ever watched this movie? Hangover, okay? And I think some of you, when you drink, you get flushed. Yeah. You get red easily, right? And that's actually because there's a mutation. Your gene, which is called alcohol dehydrogenase. And so if you carry a certain mutant in your alcohol dehydrogenase gene, then you get flush, you get red flush easily when you drink. Okay, and you also get drunk more easily, right? So this is a This is a example of how the knowledge of human genome can tell you what we can learn from that. Yeah, and so now, are you interested in learning whether you carry the mutant or not? Right? Yeah. You know, nowadays, if you want to sequence your genome, how much does it cost? How much does it cost? The sequence to know your genome, to know the mutants, the mutation, or variant, the genome.
100 to 100,000.
Not that cheap. Yeah,
$1,000.
$1,000. Yes, $1,000. I will give you.
You him too.
You don't need to.
It's okay. It's okay.
It's okay. Right. So if I ask if you want to sequence your own genome by spending $1,000, yeah, are you going to do that? Maybe.
It's an investment. Yes.
It depends on whether you have money or not.
Yeah.
Yes. So I asked my students in Taiwan, most of them say no. I rather don't know my risk for developing a certain disease. Yeah. And but if you somehow develop cancer, you have to do it. Okay. So NGS is now applicable. It's now practiced. Yeah, in many hospitals in Taiwan. Okay, especially when cancer patients they need to decide whether they want to take the targeted therapy or not. Okay, so that's one important area. Yes. Actually, I think by the way, it is very important to know how much you can drink. Actually, not in college. I think when you were in high school, and the chance of having a party is much higher. Okay, and in the party, it's unavoidable that someone will encourage you to drink, and you don't want to be wasted. Okay, so you want to know very well how much you can, especially female students. Okay, and so I usually encourage my kids, yeah, my students, they spend one day staying at home with your parents, just drink as much as you can until you pass out, and then know how much you can drink. Okay? And that knowledge is very important to keep you safe. Yeah. So yeah, when you have a holiday going back home, yeah, give that a try. Okay. This is very important. Okay. All right. Another very important application of NGS in human. Yeah. So anybody can tell. Yeah. Can tell story from the figure. This is the topic. The title is non-invasive prenatal tests. Anybody would like to try? Oh, can you recognize what this is?
Viral
You know what?
syndrome.
It's a syndrome. And it's called Down syndrome. Ever heard of Down syndrome?
Oh, now.
Down syndrome. And it has your chromosome 21 has 3 copies instead of 2. Okay. And usually the kids will grow like that. Okay. And so before the kids were born, it is important to know whether the kids is syndrome. Okay. But what's the method to check? Yeah. Before NGS, what people do is they use a needle, poke into the uterus of the mother. Yeah. And then take some and there are some babies DNA roots. Okay, and they do the sequence. Okay. And to tell, maybe not sequencing, but how they can tell whether there are 3 copies of chromosome 21 in the amniotic fluid. Okay, so that's how the potential syndrome is tested. But you know, it's kind of dangerous, right? Needle in the uterus, maybe baby like to dance, right? Yeah, some okay, so the risk of accidents doing this kind of test is also pretty high. It's like 300-500 every 1 in the 500 pregnant women. So now, with NGS, what we can do is that. Now, can you guess what we can do? Mother's blood. Okay, using mother's blood. Because the baby is connected to the mother, right? Through the umbilical cord. So baby's DNA will be also present in the mother's blood. Okay. And guess how much, what's the percentage of DNA in the blood that is from the baby? Percentage. 10%. Why are you so good? It's about 10%. 10% of baby's DNA come from the mother. 10% of mother's DNA in the blood from the baby. Okay, so we can sequence the mother's blood DNA and then figure out whether the baby has 3 copies of chromosome 20. Okay, so it's a much safer. It's called non-invasive. They don't need a needle, put the needle into the uterus anymore. Okay, so these are the important applications, and there are many other applications. For example, we can compare. Now we were talking about human genome, right? And we can also sequence the genome of different species, like for example, chimpanzee, right? So what can we learn from sequencing chimpanzee's genome? Similarities from our genome. So why do you want to find those? Huh? Find evolutionary pattern. Find an evolutionary pathway. Pattern. Evolutionary pattern. By the way, do you know how different are you from your neighbor? And so, like what I said, always carries some mutant, right? So you and your neighbors, your classmates are different, yeah, in your genome. And what's the percentage of the genome of your genome that is different from your classmates' genome? Less than 1%.
Less than 1%.
Less than 1%. Yeah. Less. How much?
0.5 or 0.7.
Mine is 0.7.
Like it really depends on your friend's ethnicity and all that because here we have different.
Yeah. But on average.
Average. Yeah. I would say maybe my guess would be wrong, but it could be around 0.5, I guess.
Smaller.
Oh, so, okay.
0.3, 0.2.
Smaller. 0.1. Okay.
Okay.
1 in 1,000 bases. Yeah. Your genome are different from your classmate. Yeah. So that's not the difference. Now, yes, the percentage of difference between a chimpanzee genome and human genome. What's the percentage of difference between a chimpanzee and us? It's about 1. Okay. Only 10 times difference, degree of difference between you and your classmates. Okay. So chimpanzee, although it looks very much different from us, that's actually very similar. You check their genome. Okay. Right. So what can you learn? You have a chimpanzee's genome at hand and your genome at hand. You learn. Uh-huh. Animals. Transmission of disease from animal. Okay. So what does it have to do with the genome, our genome, transmission of disease? Uh-huh. Okay. Okay. So HIV virus, that's virus of that's actually come from chimpanzee, right? So yeah, if you study chimpanzee genome, maybe you can have some hints on how to cure the disease. That, right? All right. So any other possibilities? Yeah, we can learn from comparing the chimp genome with ours. Yes.
We the evolutionary apa ya namanya evolutionary protection
Oh, so interesting. Family trees. Yeah, because each is interested in the history. Yeah, of all species. Right. It's indeed beautiful. Yeah, all the histories of different species. We all are, but we achieve that birds and fish are all evolved from one single common ancestor. That's amazing. Okay. So yes, we can know the evolutionary history. Okay. So after all these examples, I just want to say that so many things we can do with sequencing the genome, with sequencing the genome. And that's why so many people in the world are using NGS. Okay. Yeah, generate so much data. This and the task of a bioinformatician is to extract meaning, yeah, from the data. Okay. So it's a wonderful direction of research. You are able to extract the meaning from this ATCG, then you all can make some contributions. Yeah, many different fields, starting with interesting questions. Yeah, I just told you. Okay, so bioinformatics is quite important, and so this is. Of course, I think many of you are working on biology, microbiology, studying not only the genome of big species like chimp or dog, fish. We can also sequence the genome of bacteria. Okay. There are so many applications. Some bacteria can produce vitamin, can produce certain enzyme useful for many different reactions, right? So in order to know how to manipulate this bacteria, yeah, it is very useful if you know the genome. Okay? So that's one potential applications. And also microbes are everywhere. And then soil, there's the air, water, they are even in the ice. Okay, even in hot springs. Yeah, there are bacteria. Okay, mentioned that lots of microbes in our human body sites, all kinds of body sites. So it is very interesting to explore uh, the okay the human microbiome. So that's one of the major themes of my study. Okay, human microbiome. And I think it's already about an hour. We take a 10 minutes break. Okay, so how about we take a break? Okay, then we'll come back. I'll show you what I did. Okay. All right, let's have a break. And if you want some candy, yeah, you can come to me. Yeah. All right.
Yeah.
No?
So.
When pregnancies, it's not new in a short, it's not good. Then you can stop the pregnancy while it's still at the early stage. So it's not the project. So we can fix it because time it's not really dangerous for the others to practice. Yeah.
That's the reason is the way to know this. Yeah.
So it's that small window between the time when the time when you can sequence the baby's DNA and when when it actually started, when it actually is late to actually. But there's like a high question the Delta was before maybe I got it over
If this is the this sounds much more one second.
You can do like line in one thing like you can like give one track oh yeah.
The value of the five.
It's just one photo. Then that's something. And then at the end, it's not like it's too early to know whether the baby has.
Yeah,
Exactly. Like that when it's still the early stage where there are only a few cells. So isn't it like too early to tell whether the baby has immunization or some blood?
So usually let's like we have two checks. But we also check I pick one.
Yeah.
Pick one. Check with you.
So as I go. Yeah. The blast is taken.
Yeah.
You can do that. So you can that's very natural. Basically, almost impossible. That's very
Extract the flow. The cell. Correct.
yeah. It's really cool. It's quite we do a idea. Wait for the cells to get for example, black iris to press down certain mutation. So you can actually screen the embryo before it's implanted in the body.
Yeah, so
It's part of the
in the mountain. So when we this act
second, it's saving my limitations. But IPF and then right.
So I'll continue the topic about human microbiota because it's one of my research topic. And yeah.
Yeah, this is my thing. So in the Zoom is it's connect. Oh wait. Now I think it's fine, and then you share again.
And you share the whole screen. We share the whole screen.
It's the PowerPoint or
But I cannot share anything.
yeah, they have to share. Ya, terus kita kasih paper aja. Kasih paper kan? Kasih paper ke bapaknya. Ya, tapi maybe we use our your laptop. If you can get the Wi-Fi.
If you need the chances of the price.
So, before I jump into my research topic, I just want to give you one more last example about importance of the microbiota. Okay, so here is a nature paper experiment. Okay, very nice experiment. Can you tell what? Can you tell the meaning of this? Anyone like to explain this one? Yes.
So basically the apa namanya this apa namanya this picture is showing how your gut microbiome could affect how your body works, how your body functions. For example, like if like twins are identical genetically, right? Like but if you transplant different microbiomes into two different microbiomes into the twins, then it could affect how they absorb their nutrition uh, and ultimately it could affect how they could how obese one mouse could be compared to its twin.
Yeah, exactly. Very good. Um, so if your one can transfer the gut microbe from human to a mouse. Okay, and if you have if this mouse have microbes from this obese, okay, and then this one will end up being fat. Okay, if this one have microbes from this lean, yeah, then he will remain lean. Okay, so the summary is that if you are fat, it's your fault. Okay, it's the problem of your gut microbe. Okay, so microbes are very important, and there is something called gut brain axis. Right, so what's inside your stomach or intestine, your digestive guts, and your gastric system, yeah, will affect your brain. Okay. So if sometimes you feel blue, yeah, maybe it's because your gut microbes. Okay. And actually, recent studies have revealed that gut microbes have impacts on several neuronal diseases, including Alzheimer's disease. Okay, so learning the impact of human microbes can be very important, and this is one of my major research studies. Okay, all right. So now imagine that you have these NGS sequences. You collect micro from many different body sites. For example, your nose, your tongue, guts. By the way, do you know how to collect microbes from the gut? But know?
Stool sample?
Yeah, stool sample. You'll use stool sample to explore explore the gut microbe. Okay. Of course, you can use some endoscope. Yeah. And take samples from guts, but that's more intrusive. So usually what people do is they collect your stool of business and then try to extract the DNA and then do the so-called 16S RNA sequence. Okay, so 16S RNA is a very important long-term gene of bacteria. All bacteria contain this 16 RNA gene, but they differ in certain areas. Okay, so you can tell which bacteria it is. Okay, so with such data, the goal is to determine the composition of different microbes in each sample. Okay, right. So that's the goal. All right, and how do we do that? Yeah, explore your imagination. Imagine that you have data like this. Each single sequence comes from one particular bacteria. Okay? And you need to determine the species or genders or different taxonomies. Okay, so how do you do that? How? Discuss with your neighbors. How are you going to do that? Any potential approach? Yes.
Maybe we can use Rust from AI or something.
Yeah. Very good. Use Blast. Everyone knows Blast?
Yeah.
Right. Yeah. I have a very good question. Okay, so imagine that you have a sequence like that. To determine its species, yeah, you have a bacterial sequence. One way to do sequence alignment, okay, which is what Blast does, right? Blast is doing sequence alignment, so it aligns the reads to all the potential sequences, all the available sequences of all known bacteria in the database, okay? And they try to find out which reference sequence match the best to the reads, okay? NGS sequence. Okay, so however, this process is pretty long. Have you ever tried using it? Right, so it takes a few minutes.
Yes.
Right. So I have to wait like 20 more seconds, depending on the length of the sequence, right? And so it's very long. And imagine that you have millions of such things. Yeah. Still want to use Blast? No. And you're not going to graduate, right? Yeah. So how can we speed up procedures? That's what bioinformatics people do. Okay. Yeah, they try to invent some good methods to speed up the process of alignment, okay, or the process of determining species. Okay. So here I want to show you a very nice approach. Yeah. Instead of doing this sequence alignment, yeah, there is a tool called RDB classifier. what it does is to count the appearance, the presence of words. Words are consecutive bases in a small window. Okay. For example, this is a word CCGA, and this is also CCGA. So you do counting of words. Okay. And if you found that this NGS sequence contain words, many words that also appear in a reference sequence, then you know that they are very similar. You don't need to do alignment. And you just count. And when you have lots of similar words, yeah, you say, oh yeah, that's so it's a very simple idea, but it turned out to be very efficient. Okay, yeah, it's not too difficult, right? Just count words, and you can speed up the alignment process to 1,000 times. Okay, so this is the beauty of the challenge and the beauty of bioinformatics. Okay, all right. So after this RDB classifier was invented, people start using that to determine the species of each sequence. Okay, but still, although it's like faster by 1,000 times, it's still too slow. Still too slow. Okay. And how can you further speed up the process? How can you further speed? Okay, so it turns out that one possible solution is to first mention that you have so many sequences like this, but some of them are highly similar, and they are very likely belong to the same species. Okay, so if you are able to cluster highly similar sequences together, yeah, then you just need to annotate one of it, and the rest could be from the very likely to come from the same species. Right? So the clustering, very important procedure in bioinformatics. Okay, with the cluster sequences. And then by clustering sequences, you need to compare similarity between sequences. Yeah. And the comparison can be very difficult as well. Imagine that you have 10 sequences, and you want to do pairwise comparison. Okay, how many combinations are there for comparing 10 sequences? It's actually the problem of combinatorics. Yeah, you still remember that from your high school math class? 10 sequences. You want to pick two for comparison. How many combinations are
I wonder, like that sequences.
Yes.
There's 4 nucleotides.
four nucleotides? Each sequence is like that. Yeah, each sequence this is one sequence.
Oh no.
You want to compare 10 such sequences. It's not a single base. So how many combinations are there? Okay. In 2 from 10. That's why math is important, okay? Especially in bioinformatics. Yeah. You should not be afraid of math if you want to do bioinformatics. Okay? Yeah. So right. So let's make it easier. How many combinations are there if there are only 3 sequences? A, B, C. You want to compare the to any 2 of them. 3, yes, 3. What if you have 4 sequences? So you can pick 4. You have 4 options, right, for the first sequence, and then 3 options for the second, right? And then you can swap the 2. So 4 times 3 divided by 2. Okay, 6. Okay, so now what if you have 10 sequences? Yes, 45. Okay, that's where combinatorics works. Bioinformatics. Right, so imagine that you have a million of 6. Yeah, that's how many calculations. That's a lot. Okay, so in order to do the clustering, we need to invent another algorithm to speed up the process. Okay, and so there's an algorithm in this tool called uparse. Okay, it's able to do the clustering much faster. And I'm not going to tell the detail, but yeah, it's able to do clustering in a very fast manner. Okay. Now, once you are able to cluster highly similar sequences together, and you can reduce the number of clusters a lot. Okay. Originally, it can be like million sequences, for example, but after clustering, usually they're like thousands of clusters. Okay, so it's like thousands times faster. Okay, and with this 1,000 times faster, now your priority classifier, they can do taxonomy and notation. Then you can graduate, okay, in time, right? So that's the beauty of computational algorithms, right? So, so with this, you are able to obtain the composition of different bacteria in different samples. Okay. Now, the next question will be, are the, for example, the bacterial composition in this group different from that group? Okay, for example, if this is the microbial full microbe composition of healthy individual and these are the disease individual, then you want to figure out which bacteria are different in terms of the abundance. Okay. And then perhaps that bacterium is related to the disease. Yeah. Okay. So you want to figure out which bacteria is more. Yeah. And this simple question turned out to be not very easy. Okay. Why is not easy? There's this challenge called compositional challenge. Here is the illustration of case. Okay, so assuming that you find 3 bacteria with the same amount of abundance, with the same abundance in the sample. So you get 100 reads for this one, 100 reads for that one, 100 reads for this one. Okay, and so each of them occupy about 1/3 of the community, right? Now imagine that this bacteria expands, yeah, they duplicate faster, okay? And but these two remain the same, they don't increase, all right? And this span 2 times. So if you do sequencing, you will get like 200 reads this bacteria. Okay. Now, if you calculate the fraction of bacteria, this one will become half. This will become quarter. Now, if you compare the fraction, this bacteria, 1/3 to quarter, you'll feel that, oh, this reduced. Okay? But in fact, it doesn't change. Yeah. So there's a problem. This problem is called compositional problem because the fraction of all these bacteria should add up 1. Okay. So there's a constraint. Therefore, if you simply compare the raw fraction, then there's a problem. Yeah, it cannot reflect the absolute abundance of the bacteria. Okay, so that's a problem called compositional problem, and it's quite challenging data analysis. So, what can we do? Yeah, we do facing such a problem. Okay, and some people were genius. Yeah, they invented the approach, and the approach. The idea is to compare to study the ratio of relative abundance. These are relative abundance. Okay, and instead of simply compare the ratios of relative abundance, you can compare the ratio of two relative abundance. For example, if you compare this with that, ratio is 1. Okay? And when you compare this with that, ratio is 2. Okay? So this really increase. Yeah. But if you compare this with that, the ratio is 1. Although now the ratio is quarter, but the ratio of the two relative abundance is still 1. Okay, so the idea of comparing ratio of two different bacteria can solve the problem of compositional problem challenge of composition. Okay, so that's a fantastic idea. So now I have shown you 3 major challenges. This one, compositional problem, and this one, clustering problems and alignment problems. So in order to get this composition of microbes, you actually need to have a very nice idea about how to process the data. Okay, and that's what bioinformatics is beautiful. That's why bioinformatics is important, because they want to not only speed up the process, but also gives you the correct interpretation of the results. Okay, so these compositional problem occurs in many different ways, and you should have a good idea about that. You want to, and now that so the ideas that I just share with you, it's not too difficult, right? Yeah. And they are the kernel of these 3 different computational tools: RDB classifier, UPass, and Angle. Okay. Although there are some more detailed mathematics you need to know in order to crack the secrets of the tools, but you get the main idea of the tools. Okay. That's how these tools help people to correctly infer the composition of different communities. Okay, that's how we can arrive at the very solid conclusion. All right, so that's the fun part of learning bioinformatics. Okay, and so with these tools at hand, now I'm going to tell you some of the applications. Okay, so in this project, I collaborated with clinical doctors from the Children's Hospital, Dr. Wang Zhi. Okay, and we were interested in the nasal microbiota, the bacteria from your nose cavity, and we are specifically interested in asthmatic children. Asthma. So people have the children will feel difficult in breathing or sounds when you breathe. Okay, so we want to study their nasal microbiota. Okay, so at that time we collected samples from many different children, right? And then we extract the DNA, send it to sequencing company, okay, and they gave us some data, okay. But when we got the data back, we start to analyze the data. This is the so-called principal component analysis. So this figure shows how similar 2 samples are in terms of microbial composition. And the closer the 2 points, the more similar their microbial compositions. Okay, and then you can see that this is the blue one is the samples, the first batch of experiments. So we collect the samples from children in two different batches and send the sequencing, send the sample to sequencing company. This is the route of the first batch and the second batch. Okay, so you can see clearly that the two batches are very different. Okay, yeah, they're very different. However, the patients involved, the two batches, basically similar. They are all asthmatic children. Yeah, it's not that the first batch are all males, second batch are all females. Yeah, we have these clinical features distributed. Yeah, within the two batches. Yeah, so that's very weird. Yeah. So what do you think happened? What do you think happened? That's the real challenge you'll have when you get the data. Okay. So you expect that the two batch, the samples of two batch will be rather homogeneous. Okay. So they shouldn't be separated like here. So what might be the problem? Anyway, sequencing one sample, it costs like 5,000 new talent dollar time. Right? So this piece of data, they're like you have to spend at least like 300, yeah, $300,000, yeah, US dollars, not talent dollar. Okay. Anyway, so it's not cheap. Okay. But do you trust this data? No, right? Because they are not homogeneous. Yeah. But what are you going to do if you find this? First of all, never fully trust the result from a sequencing output. Okay, that's one important lesson. Not even from statistics, because everyone makes mistakes once in a while. Okay. And when you get the data from a sequencing company, you have to check that yourself because they're not going to check it for you. Right? Check it, and you also need to know how to check it. Right? So, for example, this is so-called principal component analysis. So if you study bioinformatics, you'll learn how to do some kind of analysis. Okay? And it's a very useful analysis. You can apply it to many different subjects. All right, so let's back to this problem. Yeah, what are you going to do? You have already spent quite some money, and you figure out that data were not consistent. What are you going to do? What are you going to do? Spend a lot of money. They are not consistent. Yeah. Sue the company. So, so what I did at that time is that I have to convince the company that they make a mistake. Okay? And you have to show evidence. Right? So here is the percentage of certain bacteria in the first batch and second batch. You can clearly see that, in many samples of the second batch, but the fraction of this bacteria is pretty high. That's almost zero for the first batch. Okay. The same for this one, same for that one. So it is possible that there is what? Keyword. Contamination. Okay. Contamination. So it is important to check contamination. Yeah. Because when you do sequencing, you actually do not sequence only one sample at a time. Nowadays, you need to sequence 96 samples at a time. Okay? So it's possible that some reads from this sample will move across to another sample. And you ended up with, if you if you, yeah, if you with some alien sequences from other samples. Okay, so that happens. That happens. So you need to know the pros and cons of NGS. For example, you need to know that 96 samples are sequenced at the same time. But you need to know why is it important. Yeah, because if you sequence multiple samples at a time, there is a chance of, okay, yeah. And so you don't just learn the facts. Oh, facts was in the same time at the same time. Yeah, but you need to know the consequence. Okay, so yeah, and because I showed that the data, yeah, they decided to return the money back.
Oh, right.
Okay, and so we use the money to do another round of sequencing, another company.
So,
Yes. Sure.
since of course, some projects or some research that needs us, demands us to sequence multiple genomes at a time. But since you said that sequencing multiple genomes at a time may cause contamination,
Right.
how do you do that? How do you sequence multiple genomes effectively without risking contamination?
Okay, so sometimes contamination is not avoidable. Sometimes. Okay, so in the field of this metagenomics, usually, okay, so first of all, what we do is we usually prepare the sample. Yeah, with known bacteria in the sample. For example, 12 bacteria only in the sample with a known proportion, each one with a known proportion. Okay, and then we do the sequence through this standard sample. Right? And then when we get a result, we analyze the number of species within this standard sample. And usually what we found is that there are 50 species instead of 12. Yeah. And then you know clearly that there are contamination. And that's not avoidable. Okay. And so one way to solve the problem is to do the so-called filtering. Yeah. So these amounts of usually those additional species, they appear in a very low fraction. Yeah. So one way to remove that is to remove the bacteria that appear with a very low fraction in the sample. Okay. So this is called terminologies to remove the crosstalk. Yeah, between samples. Yeah. So that's a very important step in the data analysis. Yeah. Especially if you want to quantify the diversity of the sample. Yeah. So we got only 12, the answer is 12, but you ended up 50. Yeah, that's a lot, right? Yeah. So you want to do some pre-process, you want to do some filtering, yeah, during the process. Yeah. And in your case, if you sequence multiple genomes, it is possible to align the reads to different genes, figure out if they are contaminated. Yeah. Okay. So what do you usually do? What a bioinformatician usually do? It is not that you just enter all the data to a tool. Just click on run. Okay. Just set whatever. No. Yeah, a highly qualified bioinformatician should know all this potential of errors. Yeah, during the precision. And you should check this, check that, to ensure a high quality analysis. Okay, so okay, so yeah, so we ask another company to do the sequencing. Now we have very confident data, yeah, to for subsequent analysis. Okay, about nasomicro. Then they are very interesting. For example, here shows you the composition of different bacteria. Okay, and this is called a heat map. So if you see lots of red, that implies that that bacteria is very abundant. So this red indicates the percentage from 0 to 100. So you can see that, so, oh, these are almost 100%. All the rest are like 0%. Okay? Yeah, so this is what we know about nasal microbiota. Okay? So what can you learn? What do you learn from this? Learn from this. You need to make good conclusion based on your feed. Okay. And what do you learn?
After asthma attack, the nasal microbiota is increased in diversity. Maybe like since we're since in an asthma attack you're likely like sneezing a lot and like coughing a lot. Maybe the some of the microbes that is in your breathing pathways, in your throat and everything like that gets pushed up to your nose. That's why there's a lot of
Okay. Very good hypothesis. Okay. And so just mention that when asthma occur. Yeah, or so actually these data are these samples were taken during the asthma attack when the children suffer from severe asthma syndrome. Okay. And these are the samples taken after they recovered. Right? So in your hypothesis, you are supposed to see something like this. Yeah. In some of the samples, you don't have one single bacteria dominating the community. You still see other bacteria. Okay? But that's not very common. Yeah. Okay? So what we learn from this one is that in our nose, usually there is only one kind of bacteria that dominate the community. For example, it can be this, Staphylococcus, it can be that, Morosera, it can be this, okay? Usually it's like one party, one party government. Yeah, it's not like in the US, like two party, but there's one exception, like these two bacteria, they somehow collaborate with each other, okay? So they can coexist in your nose, but if you added the fraction of the two, it will be almost one, okay? So the two bacteria constitute majority of the microbial community. Okay. Yeah. So this is one interesting to know that only one kind of bacteria can survive well in your nose. No, not many other. Okay. Also, interesting thing we found is that when we check the dominating bacteria and those of these children during the asthma attack. Okay, we compare the dominating bacteria with their clinical features, because some of the asthmatic children, they are allergic to many things. Okay. So we call them allergic asthma. Okay, but some other children, they are not allergic asthma. Okay, so the asthma syndrome is not related to allergy. Okay, and we found that these allergic asthma children during the asthma attack, most of the children's dominating is two. Okay, so and when the children recover, we don't see such kind of dominance. We don't see such kind of. Okay, So what does it imply? It implies that the immune system of the children is interacting with the bacteria. Yeah. But the interaction only occur during the asthma attack. Yeah. When the children recover from asthma, they behave like normal children. Yeah. Okay. So there's a very critical window of time where you can see the interaction between your immune system and the bacteria in your nose. Okay. So it's a very, yeah, in my opinion, it's like quite a nice finding. Because if you want to study the interaction system and microbes, you want to study it at the right time, right? But people used to not knowing which time best window, yeah, to study the interaction. Okay, so now we think that's a very interesting finding, and 5 minutes left. Okay, yeah, and also with that data, that piece of data, we also collaborated with a professor from Department of Measurement. Okay, she's able to predict the quality of air because those kids they live in different places. Somehow she's able to estimate the quality of the air near their houses. Okay, and then we try to see if there's any correlation, yeah, between the environments of their homes and their nasal microbiota. Okay, and it's very interesting that we found that if you live in a place with more greenness, and for example in your campus, right, and your nasal microbiota will have a higher diversity. Okay, and usually a microbial community with a high diversity is good. It's considered good because it's usually more robust, right? Yeah, you have more players, the community, then they will not fluctuate. Okay, so it's considered good if you live close to the park. Okay, and with so many trees in your campus, I believe that your muscle microbiota are pretty healthy. Okay, so that's the beauty of bioinformatics. By analyzing this bio composition, it's a very interesting, yeah, relationship between environments, between bees and the microbes. Okay. All right, so it's about time. Yeah. So I really enjoy giving the lecture here. Hopefully, you have learned something. Yeah, and remember that your career path can be full of surprise. Yeah, and so it is good to be open-minded and try to learn some informatics. It's pretty good. Yeah, and it's not so difficult. Yeah, see, I just told you some main ideas. And if you know the main ideas, those tools are not that scary anymore. Okay, so I wish that you enjoy the bioinformatics and enjoy the 2 hours speech. Okay, yeah, thank you.
Thank you for be enlightening. So now you know the purpose of why you get bioinformatics as your compulsory course. So anyway, because we unfortunately we have a very limited of time, maybe we can have a quick photo session. Yeah, would you please maybe Professor Liu here, Meta, and then we take picture together. Nas. Oh, you can sit there. Maybe yes. Let's say this one is
Okay.
sit here. Or maybe we just the left makeup.
Yes, we go. Okay.
So. Oh, middle is here.
Sorry.
Sit. Okay. We'll do. Okay, yeah. Look at that. One, two, three. One, sorry, big thumbs up, everyone. One, two, three. Oh my God. One, two. Terima kasih ya sudah semangat. Semoga movie berguna untuk kalian. Passionnya bioinformatik mau kemana karena bioinformatik ada banyak. Minggu depan kita mulai lagi dengan konsep dan teori. Oke? Sampai ketemu hari Rabu buat praktikum. Thank you.